I'm ready. This is my first time, actually, my first talk at the CRCT. So we are not that far one from each other, but I think it's really important that everybody knows what we are doing in Toulouse. And the work I'm going to present today has been done entirely at the UDR. So, and I have just put it on this picture, our new logo, Infinity, because as you know, UDR and CPTP are actually fusing. And on January 1st, we will be the Toulouse Institute for Infection and Inflammation. So I'm going to try to give you first a small overview of the peripheral nervous system. So the peripheral nervous system, and notably the sensory system, actually innervate most of the organs. So we will have, for example, the lungs that will be innervated from sensory neuron that will be coming from the nodose ganglia, from the DRG, the dorsal root ganglia, and from the para and prevertebral ganglia. There is the gastrointestinal tract that is a little bit specific because it receives projection from the DRGs, also from para and prevertebral ganglia, but he also possesses his own actually brain, his own enteric nervous system. And today we will focus on the skin because this is the major interest in my lab. And the skin, it receives basically projections mainly only from the DRGs. And we and many others since now 2013 have shown that despite their primary function in the transmission of sensation, so to induce, for example, a behavioral response like scratching or removal, those sensory neurons can also have additional crucial functions in the modulation of immune responses. And I'm going to show you how it happens in a specific disease, atopic dermatitis. So this is going to be a simplified overview of the skin neuro-immune network. So on this picture, we're going to have some dendritic cells. This is a mouse skin. We're going to have as well some macrophages. Then, as you know, in the mouse, you can find dendritic-shaped epidermal T cells. You can find dermal T cells. And on this picture, if we had, for example, the blood vessels, you see how we complexify the picture. But if we had the sensory neurons, you will find, you will easily understand that it's going to be very hard to find an immune cell that is not either in contact or in very close proximity, actually, to a sensory fiber. So one particular immune cells that I've been now studying since almost 10 years, since I was in thesis actually with Eric Espinosa in Salva Group, it's the mast cell. And you might have heard already a lot about mast cell, I guess, but I would like just to summarize a few things. We know that thanks to different knockout animals actually that are more and more efficient, that mast cells do play a role in a lot of different pathologies. They are pro-inflammatory, for example, in allergic inflammation, anaphylaxis, fibrosis, in some cancer, angiogenesis also, and they can also trigger inflammatory pain. But they can also play the role of the good guys. They can clear actually parasites from the body, especially helminths. They are important for fight against bacteria, and we recently published a paper in Immunity about that this year. They are important in the resistance against venom and for some particular tissue remodeling. They possess more than 300 activatory receptors on their surface. There is the well-known high-affinity IgE receptors that can bind monomeric IgE, and following entry of antigens into the body for which those IgE are actually specific, this receptor will be cross-linked, and it will trigger the release of preformed granule-associated mediators, such as proteases, histamine, but also embedded cytokines and preformed cytokines like TNF-alpha. We have been working during my thesis with Eric as well on some different aspects of mast cell biology, such as its capacity actually to present antigen to pre-activated T cells, such as effector memory T cells, and also to degranulate in a vectorial fashion against parasites. Today, I would like to bring to your attention actually one new receptor that has been discovered 5 years ago, named MRGPRX2. He belongs to the family of the mast-related G protein coupled receptor. It's a GPCR, and it can bind a very broad panel of different molecules. We're going to come back on it. So we developed a new method actually recently to visualize and track skin mast cell granules in living animals. And using this method, actually, we published several papers. And what we found especially is that, it's a little bit low, is that it exists actually two fundamental degranulation strategies in mast cell. And this is a paper in collaboration also with Salva and Eric again. So we found that basically if you engage the high affinity Ig receptor on the surface of mast cell, you will trigger a specific signaling pathway, and the association of specific T-snares and SNAPs proteins that will lead to the progressive release of huge granule aggregates. And those granule aggregates, they will have specific biophysical property. They will be stable enough such they will be able to traffic through lymphatics to the draining lymph nodes and kickstart the immune response thanks to the TNF-alpha they contain. On the other hand, we found that if you engage on the surface of mast cell MRGPRX2, which is the human receptor I just talked about, or its mouse ortholog MRGPRB2, those receptors are actually mast cell specific receptor. They are even more specific for mast cell than the high affinity Ig receptor. And if you engage a ligand for this receptor, and among the different ligands, they share basic properties, so they are all cationic molecules. And for example, one natural endogenous ligand is the neuro-substance P. So when you activate mast cell by the substance P, actually, you will trigger a very fast exocytosis of very small and individual granules that will diffuse instantaneously and induce a fast and local skin inflammation. So this receptor, this is extracted from a recent review we wrote actually with Steve Galley at Stanford, and this receptor, MRGPRX2 or MRGPRB2, can bind very different molecules, but they all share those capacity to have high number of positive charges on the surface. And for example, you have endogenous peptides, such as I just said, neuropeptides, substance P, the vasoactive intestinal peptides, but also antimicrobial peptides, such as beta-defensin, LL37, some other endogenous peptides. They can also bind bacterial products, and we just also published another story in 2019 in collaboration with a group of John Hopkins in cell-hosted microbes, showing that they can actually mast cell sense quorum sensing molecules from bacteria and protect against bacterial infection via this receptor. And unfortunately, they can also bind a lot of synthetic drugs and FDA-approved molecules that are all shared a tetrahydro isoquinoline motif, also named THIC. And this is also mainly responsible for injection reaction in patients. So when we engage this receptor on mast cell, you will trigger the release of granule-associated mediators, such as this I just said, but also lipid mediators and cytokine and chemokines. So the story I'm gonna tell today is about the role of mast cell and sensing of neuropeptides by these receptors, and how these receptors actually enable mast cell to crosstalk with nociceptor that produce such a neuropeptides, and in the context of allergic skin inflammation, mostly in the mouse. This work has been done by two very talented students of my lab. Nadine Serran, she's a PhD student. She just received the L'Oréal Prize for the Women in Science and for this project. And Lilian Basso, who is a senior postdoc, and he has just been recruited at INSERM actually this year in the lab. And if you want more detail, I invite you actually to look at the November issue of 2019 in Nature Immunology. So atopic dermatitis is a very frequent disease, and it's characterized by pruritus. And pruritus is very severe itching in the skin, meaning that the sensory neurons are actually activated in the patients. eczema and hypersensitivity to innocuous environmental substances, such as, for example, allergens, and painful skin lesions colonized with exotoxin-producing Staph aureus. The etiology of atopic dermatitis is really complex, like whole human disorders, basically. But what we know so far is that because of different defects in the skin barrier, the house dust mites, so those allergens that are domestic allergens that actually present on the skin of most everybody, and some exotoxin from Staph aureus will actually penetrate via the epidermis into the dermis. They will be taken up by dermal dendritic cell that will then migrate to the draining lymph nodes, carry the antigens, and actually trigger the development of a type 2 immune response that will lead to the massive production of IL-4 and IL-13, also IL-5, and production of type 2 immune response-associated immunoglobulins, such as IgE and IgG1, that will be specific from the house dust mite. And this will actually trigger the development of the pathological feature of chronic atopic eczema. There are clinical evidences that both sensory neuron and mast cells actually might play a role in atopic dermatitis in humans. If you take a serum sample or even the skin of a patient with atopic dermatitis, you will find a huge amount of cationic neuropeptide substance P. And we know that the substance P is actually only produced by a subtype of sensory neuron in the skin that have the particularity to express the ion channel TRPV1, and that it has the gene activated TAC1, and TAC1 is the gene that encodes for the precursor of the substance P. So this subpopulation, TRPV1 TAC1+, is mainly responsible for substance P production in the skin. And you will also find very elevated level of a skin mast cell protease, which is the kinase, meaning that in those patients, actually, mast cells have been activated to degranulate. And I like also to show, to talk about this case report. This is a really interesting case report of an AD patient that has actually a motor accident and a spinal cord injury. And it has suddenly healed after an injury of his sensory nerves. So basically he had atopic dermatitis all over the body. And because it becomes hemiplegic, the atopic dermatitis actually was only cleared on the legs. So to answer the question whether the mast cell and nociceptor actually would play a role in atopic dermatitis, we developed a new clinically relevant model of atopic dermatitis like inflammation. So we used two antigens that are present in most of the patient in their skin actually, which is the exotoxin SCB from Staph aureus and house dust mite G4NA. And we patched the mouse for 1 month actually with those antigens to trigger atopic dermatitis-like inflammation. So this is the kind of severe lesions in the mouse. So and you can see, as compared to vehicle-treated mice, that they develop a huge acanthosis. There is also spongiosis and infiltration of immune cells. So this is really a sick skin. And what is really important is actually that we treated the mouse with an equivalent of dupilumab. As you know, dupilumab is for so far the gold standard of atopic dermatitis treatment, actually severe AD treatment now in patient and in kids. It's a monoclonal antibody that blocks the alpha chain of the IL-4 receptor, and so it blocks signaling through IL-4 and IL-13. And we found in mouse treated with azotab control, so you have the disease that develops actually, but if you treat the mouse, you specifically block the alpha chain of the IL-4 receptor, you actually significantly reduce the development of the pathology. So meaning that this model is highly dependent of type 2 immune response actually for full development, exactly like what is observed in the patient. So we first ask a simple question. So are substance P producing neurons, so this specific subtype of TRPV1 TAC1+ neurons actually required for the development of our model? So for that, we actually first verify that the gene involved in the substance P production, TRPV1 and TAC1, were actually restricted to neuronal compartment. So this is a mapping of the gene expression of TRPV1 and TAC1. TRPV1 is here a control in different mouse tissues, and as you see, TRPV1 is actually restricted to dorsal root ganglia, so basically where the cellular body of the sensory neuron is located, and not in the immune compartment. And we observe the same thing actually for TAC1, the gene encoding the substance P. It's basically highly expressed in the DRGs and also expressed in the other nervous system, the central nervous system and the enteric nervous system. It's actually not detected in immune cells. We also used the single-cell RNA-seq databases actually to map the expression of those genes, but more specifically in different subtypes of immune cells, because it has been reported previously, proposed that some immune cells could produce substance P. And we actually found that none of them were actually able to turn on TAC1 expression, and it was only restricted to DRG. These are just specific markers actually of its subpopulation that we studied. We then confirm as well in the skin that substance P was restricted to sensory neuron. So this is a staining of substance P, and this is a pan-neuronal marker, PGP9.5, and you can see the nice overlay actually in whole-mounted skin of the staining. So substance P expression in the skin is restricted to neurons. We then actually used TAC1 deficient animals to investigate whether substance P would be required for the development of our model. These are mice treated with vehicle. So these are litter male control mice. So they possess substance P basically. So treated with vehicle or treated to develop the pathology. And we can measure a lot of different parameters, exactly like in human. We have a clinical score, so you can see the huge increase in clinical scoring here. You can measure an increase in epidermal thickness. Those mice, they have a strong infiltration in eosinophils, of neutrophils, and they produce also different specific IgG1 and IgE antibodies. If you look at their skin barrier, here is the filaggrin, an important component of the stratum corneum. You can see that this filaggrin is actually completely disorganized in the skin of atopic dermatitis, meaning that they have a completely loss of skin barrier architecture. And what was really interesting for us is that if we use a mouse that actually lack substance P, those mice were almost protected from the development of the pathology. Microscopically, so you have a decrease in clinical score, an epidermal thickness almost back to normal. They are not able actually to mount a proper type 2 immune response, no more secretion of different specific IgE, and you significantly reduce the infiltration of immune cells, and you also completely restore an appropriate skin barrier architecture. So we know that the expression of TAC1 of the substance P is actually required for the development of our model. So we next assess whether the sensory neuron subtype responsible for substance P secretion would be also required for the development of our model. So for that, we specifically deplete TRPV1 positive neurons by injecting subcutaneously and consecutively over 3 days, increasing doses of resiniferatoxin. The resiniferatoxin is a toxin that is a specific agonist of TRPV1, and it is so potent that it's actually inducing activation-induced cell death specifically into the sensory neurons. So after these treatments, we basically leave the mouse for 4 weeks, and we analyze whether the sensory neurons have been depleted just by, for example, immerging the tail of the mouse at 50 degrees. So it's not burning the tail, but it enables us actually to see whether the mouse is actually sensing heat. And if not, that means the sensory neurons have been efficiently depleted. And then we induce our atopic dermatitis-like inflammation. And compared to the MSO-treated mice that basically possess sensory neurons, those mice, they are basically white-tan mice, and they develop a very severe inflammation. Mice that lack specifically TRPV1 sensory neurons have a huge decrease of the development of the pathology. So this is decrease here in clinical score. You can see the epidermal thickness, and you can see that they are not able again to recruit efficiently immune cells in the dermis. So you also need those TRPV1 neurons in addition to the substance P for the full development of our type 2 skin inflammatory model. So we next wonder actually, because sensory neurons, their primary function is actually to detect damaging or potentially damaging stimuli and induce a behavioral response. So we actually wonder whether sensory neuron could directly detect, sense the presence of our AD antigens, the house dust mite, differentiae, or toxins from Staph aureus. And to address this question, actually, we isolate DRGs, so these are the DRGs, from the close to the spinal cord. So this is how they look like. They are actually like small lymph nodes. They look like small lymph nodes. They are not lymph nodes, of course. And this is how they look like. And then you can basically dissociate them and put the neurons in culture, incubate them with the calcium tracer, and you can put on the top different stimuli. So we're going to put the house dust mite, differentiae, but we can also put toxin from Staph aureus. So these are our 280 antigens used in our model. And we're going to put also a positive control, which is a specific agonist of TRPV1 named the capsaicin, which is an extract from the chili pepper. So in the neuron culture, actually, when you put vehicle, nothing happens. So you basically see that the calcium levels into the neurons are actually flat. This is here. And then we add differentiae, our house dust mites, and we were actually surprised to see that some of the neurons are actually lighting up, meaning that the house dust mites are actually to directly activate our sensory neurons ex vivo. And this is a calcium trace into a sensory neuron. And then we wash away in the culture actually differentiae, and then we put capsaicin. And then what we observe is that the very same neurons that are actually detecting the presence of house dust mites are the neurons that possess TRPV1 onto their surface because they are activated with capsaicin. And this is the calcium trace into one single neuron. You can see that it efficiently activates with differentiae. Then we wash away differentiae, and when we put capsaicin, we also trigger the activation afterwards. When we did the same experiments with the toxin from Staph aureus, SEB, we actually did not detect any significant activation of the sensory neurons. So we didn't find that SEB was able actually to trigger sensory neuron activation, but only our house dust mite extract. We also verify whether those house dust mites could trigger the secretion of substance P from those particular sensory neurons. So we analyzed the supernatant of culture of sensory neuron incubated with differentiae, and you can see that differentiae is actually able to trigger an efficient production of substance P from neurons, while the SEB, in accordance with our previous observation that it doesn't activate neurons, is not triggering substance P. And of course, when you engage specifically TRPV1 positive neurons, you have a huge amount of substance P that is produced. We just wanted to verify that the substance P was actually produced from TRPV1 positive neurons. So we incubated neurons from TRPV1 deficient animals, so those mice that are depleted for TRPV1 positive neurons, and we were not able to detect any more substance P secretion from cultured neurons, meaning that the house dust mites directly activate ex vivo those TRPV1 positive neurons to secrete substance P. So that was really puzzling for us, and we wonder by which mechanism actually house dust mite could be directly detected by sensory neurons. And we first characterized the proteolytic activity of house dust mites. We incubated house dust mites actually with either a cysteine protease inhibitor or serine protease inhibitor. And this is the full in blue proteolytic activity of our house dust mite extract. If you incubate with a serine protease inhibitor, you still have 100% of activity. However, when you incubate with E64, which is a specific cysteine protease inhibitor, you completely shut down the proteolytic activity of house dust mites. And this is a positive control with heated differentiae that doesn't have any more biological activity. So most of the proteolytic activity of house dust mite is actually a cysteine protease activity. And so what we did next as experiment was actually look at whether the biological activity of house dust mite extract were required for activation of neurons. So we first put actually heat-treated differentiae onto our neuron culture, and we didn't find any more activation. Then you wash away heated differentiae, you you put back normal different IA and you restore the activation. And this is just a positive control with KCL. And we observe exactly the same experiment, the same results when we specifically shut down the cysteine protease activity of different IA. No more activation of sensory neuron. Then we put back normal different IA and we restore the activation of the sensory neurons. So you need this cysteine protease activity on house dust mite to activate actually sensory neurons. And we did a final experiment. We wonder whether this would be actually a conserved mechanism of sensory neuron, of allergen sensing by sensory neuron. So we used many additional extracts of major allergens known to be involved in either atopic dermatitis or allergic asthma. And for example, we used another strain of house dust mite named deep neuronicinus. And this one is also a strain with a major cysteine protease activity, and we found that it efficiently activates the DRG neurons, while 3 other major allergens, cockroach extracts, a fungi Alternaria alternata, and a ragweed pollen with predominant serine protease activity actually did not induce significant activation of DRG neurons. So it seems that it's a property of allergen with cysteine protease activity actually to be able to trigger sensory neurons. So we know that sensory neuron can sense domestic allergen found on the skin of atopic dermatitis patients, and that if you patch a mouse with those allergens, you actually trigger a type 2 inflammation that is dependent on sensory neuron and on substance P secretion. So we next wonder actually whether mast cell would be the functional partner of those sensory neurons. So we used the first model of mast cell deficient animals, which are named KWSH mice. So those mice, they are pretty easy to use because they breed very easily. So we use them in the lab as a screening model, but they have also other abnormalities. So they are profoundly deficient in mast cell, but they also have increased blood basophils, and they have other abnormalities. For example, they lack melanocytes, so they are completely white. And here are the wild type litter may control KIT positive mice. So they develop the pathology, as you see. And when we use those mast cell deficient animals, they were also completely protected from the development of the disease. So what we like to do actually, because those mice have many other abnormalities, to use a more relevant mouse model of mast cell deficiency. So we use those mice that are named CPA3-CRE MCL1 fluxed animals. So MCL1 is an anti-apoptotic gene, as you might know. And when we specifically flux it into carboxypeptidase expressing mice, we have a mouse that profoundly lack mast cells and have a reduction in blood basophil numbers. So because these mice have a reduction in blood basophil numbers and the WSH mice have a huge increase in blood basophil numbers, you can also rule out the role of basophils or incriminate basophils by adding complementary experiment with the 2 different strains. And what we found is that those mice actually develop a severe inflammation when they are wild type. However, when those mice, when they are express the CRE actually, and are fluxed for MCL1, so when they are deficient in mast cell, they are also protected from the development of the disease. So you need mast cell also to develop our model of type 2 skin inflammation. And so, of course, we investigated the role of MRGPRB2, this new mast cell specific, it's a mast cell restricted receptor, so it's not expressed by any other immune cell actually, to see whether it would be actually involved in the sensing of substance P and the activation of the mast cell. So when we patch determined control that possess the receptor actually, those mice, you see they have an increasing clinical score, they have a huge epidermal thickness, they are basically sick. So they have an increased number of neutrophil and eosinophil in the skin, and they develop efficiently different specific type 2 immune response, and they have a dysregulation of the skin barrier. And when we used MRGPRB2 mutant mice, so those mice, they have a mutation, so the receptor is not able to signal actually inside the mast cell, and they have a totally normal number of mast cell, but just the receptor is not functional. And those mice were completely protected from the development of the pathology, exactly like mast cell deficient mice or substance P deficient animals. So there is no more increasing clinical score, they have a normal epidermal thickness, no more recruitment of immune cells, they don't have a pathogenic immune response, and you completely restore an appropriate skin barrier architecture. So you need functional MRGPRB2 to develop also this model. So because these are actually indirect suggestion that mast cell could interact with sensory neurons, we next wonder whether the AD antigens that we used in our model actually could trigger the degranulation of skin mast cells, but via the activation of the neurons. So to answer this question, we did a very simple experiment. We injected into one ear of a mouse actually vehicle, and the other here we inject a stimuli. And we analyze the readout of mast cell activation, which is the formation of an edema, because as you know, when mast cells actually degranulate, they release histamine, and this will lead to a massive blood flow extravasation locally. And when we measure ear thickness, actually, when you in the control here, when we in wild-type mice injected with vehicle, there is no edema, of course. However, when we inject our AD antigens, different IA and SCB, we see that there is actually the formation of an edema. So mast cells have been efficiently activated into, these are wild-type mice, and also wild-type mice when you inject a control isotype, in addition to our AD antigens. And when we use mice that actually lack TRPV1 positive sensory neurons, or mice that do not possess MRGPRB2, or even mice in which we specifically block substance P with an IgG monoclonal antibody, you actually completely shut down the formation of edema, meaning that we might actually interfere with a productive mast cell activation. And when you look at the face of those mast cells, you will find that indeed in wild-type mice, they have been productively activated. You see all those granules around the mast cells, meaning they really have been degranulating. While if you analyze mice that lack TRPV1 positive neurons, or in which mast cell cannot signal via MRGPRB2, those mast cells don't have a degranulated phenotype. So the AD antigens induced in our model can trigger mast cell degranulation in vivo, but for this, you actually need TRPV1 positive neurons, substance P secretion, and functional MRGPRB2 on the surface of mast cell. So these are also indirect proof actually that mast cell can interact with neurons. So we then switch to intravital imaging. So as I told you in the introduction, we actually developed a method to specifically stain intracellular mast cell granules in living animals. And we stain those mast cells into a specific mouse named PRG-CAM3 mouse. These mouse have a genetically encoded mast cell calcium tracer actually into sensory neuron. So in those mice, we can basically follow simultaneously sensory neuron activation thanks to increasing calcium flux, so increasing fluorescence, and mast cell degranulation thanks to the release of the granules. And into those mice, we injected actually a stimuli, for example, the AD antigens or any other stimuli, and we imaged the anesthetized mouse directly under a two-photon microscope. This is the type of picture we get. So if you inject vehicle, so the blue is the collagen thanks to second harmonic generation to be sure that we are actually in the dermis. In black here in autofluorescence are the hair follicles, and in red are the mast cells, and we barely see the sensory neurons because they are not activated and the fluorescent level of the calcium tracer is very low. So you see that the mast cells are actually not degranulated into those mouse. We then specifically activated TRPV1 positive neurons, and we injected capsaicin. And what we saw is that in the skin, there is actually a huge network of TRPV1 positive neurons that is lighting up in the dermis. And from those pictures, we could extract two very important informations. The first one is that most of mast cells are actually in contact or in very close proximity to sensory neurons. And the second important information is that following specific activation of those neurons with capsaicin, mast cells nearby have actually a degranulated phenotype. And what was even more interesting for us was that when we inject the SMITE antigens, our atopic dermatitis antigen, the SMITE and SCB, we found exactly the same picture: activation of sensory neurons and degranulation of adjacent mast cells. So we set up an automated computational analysis of the minimum distance between the mast cell and activated nociceptor, actually, via modeling of the fluorescence. So here is how a physical contact between a mast cell and an activated neuron actually look like in the dermis. And here it's a mast cell that you will be calculated at the close proximity to a sensory neuron, 8 micrometers, for example, of distance. And when we analyze the distribution of all the mast cells with respect to activated sensory neurons, we find that most of them are actually either in contact or at less than 35 micrometers of distance to activated sensory neurons. So they are really forming clusters, actually. So basically, it was showing that when you trigger activation of the neurons, actually adjacent mast cells are starting to degranulate. But anyway, it's not important for the main message of this topic. So to summarize, what we found is that mast cells and neuron expressing a TRPV1 and the gene for the substance P are forming what we name neuro-immune sensory cluster, capable to detect the presence of domestic allergens and house dust mite antigens actually in the skin. And this will result in the release of substance P by the neuron, activation of adjacent mast cell via the receptor MRGPRB2. And this will be the key first event that will kickstart actually the development of pathogenic type 2 immune response and the development of atopic dermatitis. So I would like to finish by thanking the people involved in these projects, and mainly Lilian Basso and Nadine Serran, but all the members of my lab as well, and also our collaborator for this work, and mainly Steve Galli from Stanford and Xingzong Dong from Johns Hopkins University, who discovered the MRGPRB2 receptor. And I would like to thank you for your attention, and I will be happy to take any question.